I really like the way my colleagues Alex Gatch noted it. I will be up here droning on about coregonine spawning habitat, but it's the thing that we're really into right now. We think we're on to something here. So I want to acknowledge my co-authors, a lot of different folks, a lot of different funding sources. Those are just some of the Cisco and Lake Whitefish eggs we're working on. So I want to start today with sort of noting our research group's objectives, which have really shifted over the past about 5 or 6 years, in focusing on which habitats and environmental conditions and the changes to those actually influence and have the largest impact on coregonine reproductive success. So that's sort of what we're trying to understand from a research perspective. In so doing, we want to do it in a way that we can quantify effectiveness of potential habitat remediations. If that is a action that the managers want to take, we want to be able to have things done in a per meter squared per cost basis to help those comparisons. Why? We actually think habitat change and its influence on reproduction, and when I talk about that, I'm talking about like 200 years of habitat change, is kind of maybe a missing piece that we haven't done a great job of understanding, a knowledge gap in those population dynamics. Sarah did a great job, as did Brian and Lucas and Alex, on understanding what's kind of been happening out there with native fishes, but you didn't notice any of them say, hey, natural reproduction's skyrocketing, it's going up, right? It's down, lower, or we can barely detect it. And so the other reason that we think this, remediating spawning habitat has a role in achieving the coregonine restoration, conservation, and restoration objectives. Also going to lead off with the take-home messages because, hopefully I provide some support for these as we work through them. The first is that there's increasing evidence for all species that when we look carefully, the spawning behavior is habitat specific. And when we look carefully, when we use gear that could detect the differences, catching eggs and where they fall, watching where larvae emerge. So not gill nets or we saw them here during spawning time, actually really drilling down and making that important inference. We've done an ecosystem experiment, so we've looked at the patterns, we see these strong sort of patterns, and when I talk about habitat, oftentimes I'm really talking about depth and substrate. Flow is in there, but those are the two main ones. And when we remediate those interstitial substrates that Lucas was identified that are really missing, we see increases, really substantial increases in egg survival. And then last, I want to just do some good due diligence of highlighting a bunch of other projects that aren't up here, and we're not going to talk about results, but I want to make the broader community aware of them so you can reach out and communicate to those researchers about it. So we really tried to shift our focus and take a habitat-specific life stage approach to the research. But what we're not working on is anything on sort of the right side of the life cycle. We're working on the left side of the life cycle in the way I have it drawn here. We're interested in processes there in the red, but we make inference about those processes by studying different stages of observation. The two key ones that I'm going to focus on here today are the egg deposition, which really is the processes of the spawning and the spawning behavior, and the larval emergence, the subsequent follow-up from those choices of what those adult fish did. Because this stuff it's not off the shelf, it's new. We've had to work with a variety of folks around the basin to figure out how to measure them. Image is a little dark there, but these metal ring egg traps simply are very heavily weighted pieces of pipe that allow us to set hundreds of these out and deploy them and retrieve them incredibly rapidly. They have exclusions from predators, and we're able to process the data very quickly. So we can get a lot of egg observations because they're incredibly variable. Similarly, our sort of Frankenstein emergence trap there with bricks and restaurant strainers and bottles and things like that is actually designed pretty carefully to have the right mesh to allow light in and really draw these positively phototactic larvae up into the trap when they're emerging off the bottom. So to come back again, if we think about this, because there's certainly debate if you read the papers about what we think coregonines have used for habitat, it often you'll hear lines like, oh, they don't use a particular substrate, or they don't care about a particular depth. That may be okay, but the more we drill in, the more we see that's probably an incorrect assumption. So if we go out there and we catch eggs and they're equally distributed in all habitats, that's sort of one end of the gradient of no habitat selection. If we go out there and find sort of what we see in the bottom, where they're actually targeting specific areas, that's sort of the other gradient. So we started working on this good 8 years ago with Matt Pavet's graduate work at Cornell, and he worked in Lake Ontario, Lake Superior, Thunder Bay, and Lake Michigan at Elk Rapids. He used a couple different gears, and what the results showed there, the black bars indicate a Cisco egg was caught. These are all Cisco in this case. A gray bar represents it was sampled, but no eggs were caught. And along the x-axis is depth, so going from 0 out to about 47 meters. In Lake Michigan, the only eggs were caught out here in plus 20 meters. Similarly, same kind of story in Lake Superior, a little bit shallower, but in Lake Ontario, dramatically different result, where all the eggs were shallow up on the rocks. Out here, they were out deep and on the silt. D is a silt-based substrate. So one of the problems we didn't understand then was it didn't make a lot of sense. Why would these species that existed here for thousands of years be putting their eggs out on substrates where they're not protected, but they're going to sit there for 5 months during the winter when there's really good egg predators out? Just didn't quite fit right. And so thankfully, folks from the Nature Conservancy, Matt Herbert, Michigan DNR, and Central Michigan University have been following up and doing more work on Elk Rapids, now with better, not with pumps, but with mats. And they find what they see the signal is very similar to what we see in Chameau, that Cisco are targeting the very tops of reefs, maybe a little bit different than Lake Whitefish, maybe a little bit deeper, but really similar. They're not putting their eggs anywhere. They're definitely not targeting deep offshore areas. The eggs do end up out there because, again, these are little 2-millimeter eggs that are almost near neutrally buoyant. So they move. They're meant to move, but they're really targeting very specific substrates. So this is just sort of we're new into this, right? So we need to be careful and not make too much inference. I think you heard in the science transfer folks about where Andy mentioned the maturity level, right? Don't sample it once and say, hey, we got this all figured out. Be a little careful with that, and we're trying to do that. But you've seen this graph now a lot from Taylor and our effort to sample all over Lake Ontario and see where we see these species. This area of Chameau Bay is just one of those areas that happens to still have a frequently successful population of Cisco and Lake Whitefish. The bubbles here indicate a larval density. So that's where we're working. That's where we've done a lot of work. These graphs show the same sort of pattern where egg deposition on the vertical axis and depth on the horizontal axis demonstrate a pattern where the egg deposition is very targeted. They're trying to put it in a certain place. Those certain places happen to often be the hard substrates covered in rocks, rocks covered in mussels and mussel hash, as Lucas noted. And the patterns are consistent year to year. If we go out in the main lake, yes, we find eggs, but we cannot find them unless we go sample in the embayments. And then subsequently, where the eggs are being targeted is where the larvae are actually emerging from. But one of the issues with that is if we want to talk about remediation, which is it? Is it the rocks or is it the depth? We don't have rocks deep. They're too correlated. We can't tease out the different pieces, right? We have bedrock and cobble and mussel, rubble, and hash, shallow, but out deep it's sand and then transitions to a lot of silt. So which one is it? That's where the ecosystem experiment comes in. Test if by stocking rocks, if we can add interstitial substrates at these different depths, can we improve egg survival? This is a great image. Thank you, Fish and Wildlife Service, Dimitri Gorski and Joe Johnson, for bringing the multi-beam technology and equipment. You know, we wouldn't be able to make these kinds of great scans. This is Middle Shoal, in a section of Middle Shoal in Chameau Bay, and these are our reefs. Throw it in parentheses, maybe more rock piles is what you might call them, especially if you're familiar with Coreon and some of the reefs and things being built out here. But these are our 4 substrate addition sites, and then we have 4 subsequent control sites. And this moves anywhere from about 1.5 to 2 meters down to about 9 meters. So that's the gradient we're trying to span here. And so we'll put egg traps out there, 50 egg traps per site in November, pull them in December, have to be ready to work right before Christmas, because a lot of times these fish aren't dropping eggs till the second week of December. And then you have to be ready right away. All the traps are out. They go out as soon as the ice is out, as soon as we can get safe access, and then we run those traps through May to see where the larvae are emerging. So the first year it was maybe a little disheartening. We put our gear out, and the red here represents the substrate added sites, and the white and black box plots represents the control sites. And we had significantly higher egg deposition on the control sites. The fish were avoiding those areas. If you're familiar with, a host of lake trout studies where they looked at what happened after you put the reefs down, people say it either took a while, or the fish didn't like it, or it didn't smell right, or something was happening there. So we were kind of like maybe expecting this, but we were sort of amazed it was that low, that they really were avoiding these sites. But equally enough, we were that surprised when we went out and we measured egg emergence on the y-axis and time on the horizontal axis. Through time, egg emergence was unbelievably higher at these control sites, despite that much, much lower deposition, right? So a control site had barely any, there was some, but not very much emergence. And so you think about what would happen naturally in that year, all that deposition goes to nothing. Very few larvae are produced. On our experimental sites, we actually have quite a substantial amount of larvae produced. So by having the deposition, how many went down per meter squared? Having the emergence, how many come up per meter squared? We can calculate egg survival. And this is just sort of the combination, right? The ratio of those two metrics across different depths for our control sites, egg survival that's less than 1%, and then egg survival in our substrate added sites that's anywhere from about 10 to 20%. So anywhere from 30 to 200%, or 200 times, higher than what we have control. We actually, even lake whitefish, everybody's been noting, we do the best we can, but there just aren't many of them out there. But we did get enough larvae and we did get enough egg deposition to be able to calculate values just at these 3 areas and see that we see the exact same result in Cisco as we see in whitefish. So we are interested in not only the sort of the fish response, but also the hydro and geomorphic response. This is what it looked like before we dropped the rocks. This is what it looks like after we drop the rocks. You know, very extreme difference in the amount of interstices. But this is what it looks like at the rocks a year later in the shallow areas and a year later in the deep areas. So we continually scan these with the multi-beam to get a digital representation of what's happening to them, and we continually drop these cameras and quantify the biotic response, and the cladophora or cladophora-like paraphytic algae, and understand how quickly these things are going to fill in. If a spawning bed only fills in every 2 years, what's the value to the population? Or if it takes 20 years to fill in, a lot different decision making. Okay, so other projects. While it's great to be working out in the field, one of the things we can't do is represent mechanisms or represent gradients fully. We can't allow mechanisms to compete and really test them like we can in the lab. So this is some work that Alex Gatch and others are leading at our Cortland station, our field station, Tunison in New York. It's really shifting from fish hatchery to a research station. And it's some neat stuff where we're just testing how different substrates, no substrate, sand, and rock influences egg survival once we start adding turbulence, right? We know these fish, we know they spawn in late December. If there's no ice, they're getting moved around a lot. How does that influence survival? Similarly, we're thinking about substrates with different organic content, be that silt, shell hash, and controls, and incubating those eggs throughout their normal temperature cycle to understand how it influences survival. Please reach out to Alex and others on that. Through a lot of the lessons that Dr. Ed Roseman and others taught us, and Bruce Manny from the Huron-Erie Corridor work, you got to get the hydrodynamics in there too to understand the erosional processes and the depositional processes. So we paired with University of Buffalo researchers, Dr. Joe Atkinson, Zendu Zhu, and Trevor Krabenhoff, and we're trying to downscale their hydrodynamic models for Lake Ontario into these bays. So really take them from the big scale down. It's working. It's not easy, but it's working. But we are getting things like, one of the reasons it's tough is because the water currents are just a lot lower magnitude than they're used to moving around in the main lake. So we have velocities of anywhere from 0.1 to 10.5 centimeters per second. So slow, but nonetheless, there's really neat areas where the flow on the surface in the blue and the flow down deep are opposite. They tend to be where the fish are spawning. So we're trying to incorporate this idea of flow into our understanding of what's influencing both the fish behavior and the subsequent survival. Then we started talking about bloater, right? And one of the questions, even though we're kind of moving down the road in restoration, is, does Lake Ontario still have the substrate and the conditions we need for bloater to survive? That begs the question, all right, what are those conditions, right? We have some idea, we have some inference from catches of bloater larvae in Lake Huron and Lake Michigan, and some catches from commercial fishers on where we think these fish aggregate, but they can be caught anywhere, and the bottom type can be pretty patchy and different. So this is some great work that just happened last week where Alex Gatch and others, this is GLRI coregonine template funded work, worked with the folks from UW Milwaukee and the Niske. Thank you for having a boat that can work in the winter. And we're out at the Mid-Lake Reef in Lake Michigan, filming a lot of lake trout and lake whitefish, no bloater, right? Unfortunately. Things aren't working out perfectly, but we're going to dial it in. This is a pilot year, but using things like acoustics to try to identify where these fish are, and then the folks with the really high-resolution ROVs to understand what's down there. And then, in addition, the folks at Ontario's hatchery, White Lake, Tim Drew and Kyle Reynolds, had a banner year of egg production this year. So we have this opportunity to actually try some bloater egg translocation. We're going to hopefully go out next week, drop eggs down into cobble and mussel substrates and sand, and then test that emergence like we've been doing in the other systems. So it seems to be important, and we feel like we're onto something, but one thing I wanted to understand is kind of look back into history, right? Like there's a lot of things in the Great Lakes where you can learn so much from the historical perspective. And I found this paper, Fish Commission Tech Report 41 by Frank Egerton, and if folks are interested, I'd encourage you to look it up, where it's sort of a social ecological case history of what science and management was thinking the issue was with the Lake Erie Cisco collapse. John Van Oosten founded the lab I work in now, focused very much from the fishers and sort of a stock recruitment approach, whereas Thomas Langlois, I think they referred to him as Hucks, if anybody knows anything about it, tell me more about it. He was an Ohio State professor and starting the Stone Lab and was working out here and living and observing the Eastern Basin. And what he was calling pollution was really incredible amounts of sediment coming into this portion of the bay, and noticed the fish weren't using and weren't spawning in those areas anymore. So just something interesting to think about, and just sort of the perspectives on where and how we study this stuff. I keep this in here, Alan's great paper in PNAS where he totaled up the cumulative stressors across the basin. And you know, we often work in one area or one region, but we don't get a lot of diversity in where we work. So it's real easy to think if you're working down here, it's just the way things are, right? But we get into this shifting baseline where if you don't see what things look like up here, you don't maybe understand how degraded they are. And we're talking about these slow to change ecosystem processes over what's a career, 20, 25 years. These things aren't changing dramatically enough, but when you step back 200 years or substitute space for time and go to other lakes, you see things are dramatically different. And so that's just a perspective that we think we try to keep in mind when we're working in any of these lakes, and especially sort of anything from Lake Erie on down. So with that, I think Mike's telling me we're out of time, but thank you. If you have any questions, please reach out. We'll go from there.